Welcome to the X-ray lab of the Fraunhofer IKTS. My name is Jürgen and I will show you our X-ray nanotomography tool. The nanotomography tool is an X-ray microscope which makes enlarged images from a tiny sample with a very high resolution up to 50 nanometers. And I will show you what it consists of. There we have a rotating anode. This is our photon source. And the light goes through the illumination optics to the sample, which is here on this stage. Afterwards follows the Fresnel zone plates, which project an enlarged image to the detector, which is then filmed by a CCD camera. Our samples are quite tiny, the width of a hair approximately, and typically are put on top of a needle. However, if we have such a tiny sample, we can image it non-destructively. This means we can have the 3D shape and the inside of the sample without destroying it. And I will show you some application examples, what we are going to do with it. So, at first I will start with the ceramic composite material. We have a ceramic fiber composite material. From a large sample, we take a small area, a small fraction. We have the radiographs in absorption contrast, and the more dense the material is, the darker the image is. After taking a tomography by rotating the sample by 180 degrees and taking a lot of images, and then a reconstruction by filtered back projection, we have a 3D data set where we can do arbitrary cross sections in different directions. So we can look at the fiber cross section, we can see the different phases inside the matrix and the fibers here in this example. And we then can apply different imaging analysis methods to highlight the pore distribution around the fiber, or also to look for inclusions here in bright. But what we have to a large extent in our group for micro and nano analysis are microelectronic samples. And modern microelectronics consists sometimes of several dies stacked on top of each other with interconnect structures called the through silicon vias, sometimes with filling defects that we want to visualize, and they are soldered together by small solder bumps. We can image those and then do a segmentation and highlight the different phases. And we can combine images from our X-ray microscope also with images from SEM to have elemental analysis. But we can also look at quite large samples if we don't need a 3D information in total. For example, for MEMS devices with such a COM structure, we can do a stereography imaging, so looking at slightly different angles and then calculating the depth information for the COM structure, for example, from the image location. And because we have a little bit space between our optics and the sample, we can also put a little bit larger sample stages inside, like this miniaturized micro DCB stage, where we can pull a sample apart. The sample in this case consists of a microelectronic device with a back end of line structure, so all the metal lines, and a dummy wafer on the back side. And with this, we can pull the two cantilever parts away from each other and drive a crack to the sample. And by taking a tomography at each opening step, we can follow the crack evolution. So a crack starts from the left, then here it's just before the crack stop structure, and then in the end this crack stop structure breaks. With this method we are able to investigate how microelectronic devices, for example in harsh environments, can survive mechanical stresses. Another in situ stage we have is our indenter. There we have a sample from the bottom, and from top there will be an indenter made from diamond. Could be an indentation tip, or could be a flat punch. Or we can also flip the actuator and do pull experiments inside. What we recently also have a lot in our group is the investigation of electrode materials from batteries. And if you have a tomography with all those details from the spherical cathode particles, we can also do image analysis and look at the pore structure, the pore network. We can also look at the distribution of the size from the individual particles and can get material data. We also can use the 3D data to segment and divide them to individual particles, here in false color. Every color presents an own particle. And then we can have a look at the individual particles and get quantitative data for each particle. That could be the volume, that could be the surface, that could be the center of gravity, you name it, what you want. We can also look at the properties of the assembly of different particles. So where do they touch? How many border voxels do we have? How many pores and cracks do we see in the particles? And last but not least, I want to show you our heating chamber. We can also put this small oven within the microscope and can do a tomographic recording of the sample through this tiny hole here. And then under a temperature up to 700 degrees C, we can, for example, look at membranes from a molten carbonate fuel cell. And there we see the different phases in different grays. So the electrolyte is here dark gray, and the white matter is the nickel oxide from the electrode. And we combine this data with lower resolution data from micro CT, and all together we can form data for simulation, so for FEM simulation in the end. This was my short introduction to X-ray microscopy in the Fraunhofer IKTS. I hope you enjoyed it, and goodbye.